are. How many of you are molecular biologists? Okay, organic chemists or chemists, biochemists. Okay, what the hell are you? Say loud.
A mixture. I think
All different areas? Can we name two of them? Zoology? Biomed? Biochemistry. Okay, cool. What? And you? Biotechnology. You? You? Biochemistry. Okay, cool. So it is a good mix. So everyone's had some chemistry. Everyone's had some molecular biology. Has anyone had physics? Some physics? Excellent. Okay. Mathematics, there's statistics? Little? Okay. No? Some? All right. That's fine. That way I know sort of vaguely how I'm targeting here. Okay, so we're going to start out with a little bit of a historical perspective. So DNA sequencing was a costly and very expensive technology. It goes back to the 1970s, some of the basic concepts, where folks were doing a whole PhD to sequence one RNA for one gene, right? I mean, it was a massive undertaking to learn the sequence of biomolecules early on. And it was a very, very slow climb between the 70s and the late 80s without a lot of technological evolution. But there were some very bold, very daring people in the government, the University of Washington, Cold Spring Harbor, and elsewhere, the Lawrence Berkeley National Laboratory in California, who had an incredible vision that instead of slogging through one gene at a time, 4 years to get one gene, let's sequence the human genome. It would be one of the largest scientific undertakings ever done. It cost billions of dollars before it was complete, but they set out to do it. And that created a massive pool of money that industry could now pursue to develop technology to make more money from this endeavor. And of course, the wonderful technology of Sanger sequencing, chain termination sequencing. You guys know how all this stuff works, right? A little bit? No? Kinda? Okay. So you had a nucleotide, a labeled nucleotide, that would terminate replication. So it would replicate up until it hit that nucleotide. So what does that mean? That means if you take DNA, load it with one nucleotide, and then give it polymerase, you're going to get it to terminate at precisely a certain nucleotide, which is wherever you stopped it. So that gives you a gel you can read off a sequence. So this was still pretty darn slow way and tedious way to sequence something that has at least 3 billion nucleotides. So whole genome shotgun sequencing was, of course, the first massive breakthrough from Craig Venter and others. And this was the notion that you take DNA, you fragment it into big, real pretty darn big fragments. And then you clone those into plasmids and you amplify them in E. coli. So now you've got great big segments of DNA that have a very large copy number. And you can now take those plasmids, you do a plasmid isolation. Who's done a plasmid prep? Anyone? Some of you? Okay. You do a plasmid prep, you isolate the plasmid, and then you start working on sequencing those. And that was nice because you knew that you were sequencing precisely one plasmid, and so you started to get these pretty long reads, these pretty long contigs that you could begin to try to assemble. This also created a computational challenge. Remember, probably none of you guys were alive in the 90s, but no, of course you were. I'm bad at math. Yeah. Okay, so in 90s, computers were terrible. They were slow, clunky, awful things. They had no memory. So it's created a computational as well as a scientific challenge. So the idea was to take this technology, as clunky as it was, and automate it. And so a few massive rooms, this one in at the NISC Center in Rockville, Maryland, and just start to do this massively in parallel. And so this was scaled up enormously, and they managed to get a whole lot of sequencing done. This was, of course, incredibly time-consuming, very expensive, and it relied on colony propagation. One really interesting thing, there were lots of blacked-out zones in the human genome because you could not amplify it in E. coli. The gene product, whatever it was completely lethal. We now know that most of these were disordered proteins, especially RNA-binding proteins with large disordered domains that are presumably stabilized by binding to RNA molecules. But we still don't bloody know that, right? 2017, fast forward. We still don't know why. But there's a lot of sequences you absolutely can't clone in E. coli or most any other bacteria. They just create sticky plaques and kill the daylights out of them. So that sucked. Now the first big breakthroughs were these 454 and other much higher throughput sequencing technologies, and then eventually came Illumina, which started life, of course, here as Solexa, and these increased the capabilities by many orders of magnitude. All right, so and those technologies, I think we'll have a few slides on them in a bit. But you can read about them online too. Illumina has wonderful videos of how their technology works on the website. Who anticipates they're going to be using sequencing in their career? Anyone? Some? I would suggest every single one of you if you're going to be within a mile of biology. Yeah. All right. So this is an old plot. Many of you have probably seen this before. Cost of raw megabase of DNA sequence. Back in the Human Genome Project, 10 grand to get across a megabase. And as of this, it was about 10 cents. And now it's about a tenth of that. It's about a penny. So that's good, right? There's lots of improvement, continuing to fall. But this is no longer dropping. We're well outside of this exponential regime. This exponential regime is really the dawn of Solexa and Illumina. And it's plateaued since then. Part of the reason it's plateaued is because we've had an almost monopolistic structure. A couple companies that really control sequencing and not a lot of motivation to cut costs, sadly. So it's progressing. University of Birmingham has lots of wonderful sequencing cores. You can get your hands on just about every type of sequencing here. From a good old-fashioned Sanger sequencer, which is wonderful. Sanger sequencer, you give it many clones of exactly one molecule, and you can get a read that's quite long. But of course, you're literally doing one molecule at a time, one cDNA, one couple KB piece of DNA, whatever you like. So let's go through the big guy. Illumina. This is certainly still the dominant entity on the block. It's a $27 billion enterprise, and it is the basis of the vast majority of functional assays and functional genomics. For a long time, Illumina was used to sequence genomes. That's changing now. You still use it some to do genome resequencing, but there are now technologies that provide much longer reads, make life much easier. So in Illumina, the basic idea is that you break things up into small molecules. You stick adapters on, you ligate them to a plate, you clone them in place so that you have many copies, and then you do more or less the old terminating trick, but with essentially much higher spatial resolution. So these are adapters stuck to a sequence. These are them getting ligated to a plate, and the clever bit is you then loop them over next to a complementary primer, and then from this purple primer, you start to replicate back the other way toward the pink primer. And you can redo that process many times. So you can amplify in place these big clusters, right, nanometer across clusters of identical DNA molecules. And that's extremely advantageous because it means that instead of trying to capture a single event, you capture many. Now, when Solexa first developed this technology, they thought, we're going to do single molecule sequencing. We're going to do one molecule at a time. And they tried it. They had absolutely no signal, right? You didn't have a fluorescent probe that was bright enough to unambiguously capture a single molecule. The huge breakthrough was when they realized they could create these little forests of identical molecules spatially localized in the same spot on the plate and then do them all at once. So instead of trying to capture a single fluorophore, they now have this huge cluster of, say, 10,000 fluorophores that lights up in a bright dot. Has anyone ever seen a movie of an Illumina chip actually doing sequencing? It's very pretty. Yeah, it looks twinkly lights, because as it goes. It's very nice. In California, we have a hell of a lot of earthquakes. And every time there's a 4-pointer or better, you have to completely recalibrate the camera. Because, of course, you're asking for nanometer resolution. It's pretty entertaining. Okay. So this reversible diterminator sequencing by synthesis stuff is pretty cool. And it has made Illumina an enormous amount of money. It's also enabled some pretty incredible assays because now you have a way And so that because you're localizing these things on a chip and then just basically taking a movie with extremely high signal-to-noise ratio, Illumina gives you a source of incredibly high fidelity base calls, right? 99.99% of your bases are going to be accurate. It's amazing. So anytime you can take a genome, chop it up, grab some of it, sequence it, and answer a biological question, you make Illumina a lot of money, and you get very, very clean results. So who knows the assay ChIP-seq, chromatin immunoprecipitation sequencing? So it's a nice application where you take a protein that you're interested in, where it binds to chromatin. You want to know where this sucker binds along the genome. Maybe it's a transcription factor. Maybe it's a histone modifying complex, whatever. You have an antibody to it. You put the antibody, you fragment the genome, you pull down all the fragments that bound your protein of interest, and you sequence them. And now you have a map, peaks of sequence matching along the genome. You can see everywhere the bloody thing bound. It's been incredibly powerful. So Ion Torrent was the next completely orthogonal technology to come out. So instead of using these Terminator sorts of approaches, well, I mean, this is similar, but instead of going after light, going after a fluorophore or something visual, this utilized semiconductor technology. They trapped DNA molecules in little hairballs at the bottom of wells on a semiconducting chip. And when a base is added, when a nucleotide is added, releases a hydrogen ion. And detecting one hydrogen ion, total bastard, forget about it. But if you do the same thing Illumina did and you figure out a way to amplify stuff in place, which they did, then you can do a lot in parallel. So you add one base, you get a whole bunch of hydrogen ions. What does that create? It creates a huge change in the pH of an ionic solution in the well. That is electrically detectable. So now you have a change in electrical current every time a nucleotide is added. So you can do the same old trick that goes all the way back to the 70s. You wash one nucleotide over at a time. You see which wells have a change in electrical current. You keep doing that, you read off the sequence. This is incredibly fast, very cheap, and extremely easy to call nucleotides, not very computationally intensive. With Illumina, of course, you have expensive and huge video files. These files are much smaller. It's just, electrical currents well by well. So it's pretty cool. But they haven't done a ton because they have not been able to track pricing with Illumina. So they may fall into obscurity. We'll have to see. But they've been very interesting for a while, and it's been nice to have a completely orthogonal source, right? Because one would like validation. You sequence your DNA on one platform, but if you want to be absolutely sure, it's nice to check with a completely orthogonal technology. That sounds good, right? Sounds good. You think anyone ever actually did that in real life? No, of course not. Right? We're cheap. You don't have enough money necessarily to do that, but it was a nice idea. Okay, so then this just shows a graph of the nice trick they do with the hydrogen ion release and the voltage detection. So each sequencing modality that has come along has a slightly different error profile in terms of the kinds of mistakes that it makes when sequencing DNA. And this turns out to be pretty important, actually, because different types of errors make it more or less challenging to understand what the underlying sequence actually is. So when you're sequencing DNA, you're doing one of two things usually. You're going to try and assemble the fragments that you sequence into a genome, into a reference genome, or you're going to map the fragments back to a reference genome. That's called genome resequencing, or that's what you do with a functional assay, like with ChIP-seq, right? You want to see where the peaks are along the reference genome. And if you've got substitution errors, where a base that should have been an A is reported as a C, what is the computational search space for that? It's linear, right? If base substitution is your only problem, then you got to check all four bases, right? 4 operations. That's not so bad. Substitution errors are pretty easy to handle. In fact, you can have one base in 10 flipped and still usually figure out where the hell the thing came from. Okay, homopolymer errors. These are a predictable type of deletion that the 454 platform suffered from a lot, which is just one of the earlier next-gen sequencing platforms. And this is where it gets confused when it's got one nucleotide many, many times. So if you have shown there 4 T's in a row, maybe it thought there were 3. It's just a side effect of how the sequencing modality works. If you have 20 T's in a row, maybe it thought there were 10, can create more problems. But the thing is, because it's predictable and you know it only happens at homopolymers, you only have to check for deletions of this kind at homopolymers, which is nice. And if you have a reference genome, you already know that. You're not too worried. So those are certainly more challenging than substitution errors, because if you've got a gap and now you have to do gapped alignment, what's the search space for that? It's exponential, because you don't know how long the gap was. You don't know how long you have to extend it. And so you end up having to check a potentially exponentially growing number of challenges. So one generally likes these SNP type errors, single nucleotide polymorphism type errors, much better than one likes insertions and deletions. So let's talk a little bit more about ChIP-seq. So this was one of the earliest new, completely novel applications of next-generation sequencing. But of course, we already had, it wasn't conceptually new. Chromatin immunoprecipitation goes way, 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 way back. First time someone had an antibody for a DNA binding protein, they thought, oh, wow, let's try and pull it out of solution. And of course, people were doing ChIP-ChIP with Affy microarrays. Who remembers microarrays? Yeah, right? Some of you? Yeah. People were doing ChIP-ChIP on microarrays going way back. This is where you had a tiling array of sequences, and you did everything by hybridization. But with ChIP-seq, now you could get much, much, much higher resolution than you ever could on the old arrays as to exactly where a protein bound. So you have an antibody to the protein, you pull it out, you sequence everything you rip down, usually on an aluminum machine, you map them to the genome, you get reads stacking up. This is literally a visualization of the raw reads mapped to the genome. And they show you a nice tight little peek over the first exon of the transcription factor NeuroD1. It's pretty cool. Okay, and so this could be done in increasingly high throughput. People can now do ChIP-seq in many, many parallel wells. So hundreds and hundreds of chromatin binding proteins have now been surveyed. In human, we've now got ChIP-seq data in at least 2 cell lines for more than half the transcription factors in the genome. That's pretty cool. Anyone know how many transcription factors there are in human? Depending on who you ask, about 1,700, something like that. And as many, again, RNA binding proteins. So this is a pretty big number. But of course, having it in 2 cell lines isn't saying much, right? Those are 2 very specific cell types. They've both been in culture for a long time, so they're pretty derived. They're a human-derived system, but they left the human race behind long, long ago. K562 has 41 chromosomes, one of the 2 main cell lines used for this kind of work. Not brilliant. But in at least one cell type, we've seen a hell of a lot of these proteins, and now we've got antibodies for many of them. Some of the antibodies are durable resources, some of them are not. Does anyone know the difference between a polyclonal and a monoclonal antibody? Okay, so a polyclonal antibody is something that you isolate from a critter, whatever, but it's a collection of cells that produce this antibody. It's not just one cell. Monoclonal antibody is where you have exactly one cell lineage. You can amplify it forever, and it's exactly one antibody, right? So it's got one binding site in the protein. The huge advantage of monoclonal antibodies is that they are forever. You have it, you can keep it, you can amplify it, it's a durable resource. Polyclonal work better for ChIP-seq because you've generally got antibodies to many different sites on the protein surface, and that can create more reliable immunoprecipitations, but of course they're not durable because you don't have an indefinitely clonable resource. You have a batch. You have a batch of antibodies. Okay, RNA sequencing. So the first huge RNA-seq paper was published by Ali Mortazavi and Barbara Wald at Caltech in 2007 or 2008, I guess it finally came out. And then lots of other papers came out in many, many organisms. So RNA sequencing really revolutionized how we think about genomes because for the first time we could sequence every RNA molecule in a cell, or do our very best to anyway. Sequence every RNA molecule. And so this is, light years beyond the 70s, where it took 4 years to get one transcript for one gene. Now we're talking about taking all of the RNA in a cell, bulk isolating the RNA, reverse transcribing it into DNA, fragmenting it, and sequencing it. So this has many, many massive advantages, right? No clonal problems with E. coli. For the first time, you can see everything instead of just a partial picture. And we immediately learned that the transcriptomes of virtually everything were radically more complicated than we had ever supposed. It turned out that with the legacy technologies, for the most part, we had been surveying highly expressed protein coding genes. Now, all all of a sudden we see that almost every protein-coding gene has bidirectional transcription from its promoter. There's antisense transcripts everywhere. There are tons and tons of non-coding transcripts. The transcriptome is enormously complicated. If you do a good nuclear isolation prep and take just the nucleus of a metazoan cell, I don't care whether it's a Daphnia, Drosophila, zebrafish, human, mouse, you name it, you are going to see just about every nucleotide in the genome transcribed at one time or another. Transcription is an enormously important part of not just gene expression, but chromatin organization, right? Getting something ready to be transcribed by a giant complex like polymerase is a big deal. It can be used for chromatin organization. So everything is transcribed. And so now the question is, how do you annotate genomes, right? How deep down that rabbit hole do you go? Here are the good old-fashioned protein-coding genes we've had computational predictions for since 2001. This work was done circa 2011. And now we know all of these antisense transcripts exist. We've got beautiful assays like CAGE, which capture the first nucleotide, the first 27 nucleotides of every transcript, so we know exactly where they are. Who knows CAP? Does that word, CAP, 7-methylguanosine? So on the 5' end, transcripts have a modification to keep them from being eaten up by exonucleases. Okay, yeah. So cap trapping was invented by a guy named Piero Carnici when he was a postdoc in Italy. He won the Italian National Medal for Science for it. It's absolutely incredible work. He developed this technology when he was a kid, and it just revolutionized everything because for the first time we had 5' complete libraries. Right before Piero, almost all genome annotations started with the first base of the MET with the start codon, right? Because we could computationally identify where the protein coding sequence began. We could annotate that. But the untranslated region, the 5' untranslated region in the promoter, who the hell knew? And so the first CAGE libraries, the first major cDNA libraries, which memory serves were done in Drosophila, gave us the 5' UTRs, and now we had 5' complete transcripts, which was really cool. But now, so we've got all this flotsam and jetsam, all of these bidirectional antisense transcripts, long non-coding RNAs. Who's heard long non-coding RNAs? Is that popular? Pretty interesting stuff. But it was unclear in general how much of the genome should be annotated. And one consortium called AceView, shortly after RNA-seq, annotated 110,000 genes in the human genome. When you think human genome, how many genes do you think? Come on, some of you know this. Loud and proud. How many genes in the human genome? 20K. 20,000 protein-coding genes in the human genome. Right, but there's microRNAs, there's lots of other types of functional RNA-coding genes that we know exist, but then all this other stuff that is transcribed potentially at low levels, what do you do? Is it a gene? Is it not a gene? Well, it's been highly debated. A lot of folks say, okay, there's another 20,000 RNA genes. Some say 40,000. But it's unclear because it's unclear how much of it is functional. There are long non-coding RNAs that are positionally conserved across all metazoans, pan-metazoan conservation. What do I mean by positionally conserved? I mean, here's a protein-coding gene, here's a protein-coding gene. They're in synteny across metazoa. And there's a long non-coding RNA between them that is transcribed in every critter. Right? I don't care if you're a sea urchin, an insect, a crustacean, a human, I don't care, a fish, it's there. What are those? Are they functionally important? In 2000 bets, functionally important? Yeah? Yeah, probably, right? So the fantastic study by Welcome Bender in 2015-2016, he took the very best known one of these guys, IAB8, or BXD. BXD in Drosophila, it's in the Hawks cluster. And he took this sucker and he left the promoter intact, but deleted all of the sequence that was normally transcribed. No phenotype. He took the promoter, switched the direction, so it transcribed the other way. No phenotype. He deleted the promoter very precisely so it wouldn't be transcribed. Phenotype. Phenotype. Well-known phenotype. So that's weird. So what is it? Why is it transcribed? The sequence being transcribed doesn't matter at all. Or it appears not to matter. But the act of transcription is important. Right? It's actually larval lethal if it's not transcribed at all. There's an experiment that's even earlier than that and even more mysterious by David Hausler at UCSC. He took a 1.8 megabase gene desert, we thought at the time. This was before RNA-seq. This is, I can't remember, 2003, something like that. 1.8 megabase gene desert that's conserved between mouse and human across all mammals. The incredible island of conservation, yet no genes. Deleted the whole damn thing. Couldn't find a single phenotype. He measured 17 different phenotypes for the mice, pretty quantitatively with a good number of mice, good number of litters, and he couldn't find a phenotype. There was a very, very slight phenotype, but he decided it wasn't statistically significant due to multiple testing. But there was a very slight phenotype for fertility. We knew after RNA-seq that gene desert had 11 long non-coding RNAs in it, 11. But every single one was expressed only in the male germline. So that's kind of cool. So if he'd looked a little bit harder, had a few more mice, that p-value maybe would have shown up. Maybe there is a fertility, a paternal fertility phenotype there. So that one of the challenges was saying, okay, this is functional, this isn't functional, is that you never know what you're looking for. Right? Not all phenotypes are lethality. Some phenotypes may be a slight loss of competitiveness in your natural environment. If something makes a fruit fly marginally less attractive to the opposite sex, that can be totally, selective sweep and then some, right? Gone, forget about it. In the wild. But in a lab, you're never going to know. You got 20 identical flies in a vial. How are you going to find out? So phenotypic studies in declaring what's functional and what's not have been tough. And as a result, genomes have tended toward the condition of annotating every goddamn thing under the sun. My group provided an annotation for Drosophila in 2014, and some people it's driven them bloody nuts, other people like it, right? Because you see everything that's there, but you've got now twice the number of genes you're used to. Most of them have no annotated protein coding sequence, et cetera, et cetera, et cetera. Another challenge is that only recently have we had a sufficiently large number of species to identify conserved sequences that are quite short. Right? Computational models that look for conserved sequence in multiple species alignment rely on A, good anchor points, so you got big long regions of solid conservation, a la protein coding genes, right? A la ORFs. And then you've got stuff that's pretty nice and alignable over reasonable stretches. What if you got a short ORF that is 11 nucleotides long, 12 nucleotides long, helps if I can divide by 3, 12 nucleotides long, you got third base wobble, right? So of those 12 bases, 4 are going to be completely unconserved. So you got 8 conserved nucleotides. How the hell are you going to find that? How deep do you have to sequence to see that? How many species do you have to see that? If you sequence enough of the same critter, like thousands of lines of a fruit fly, you could detect it statistically because you'd have single nucleotide wobble at the codons and not the other bases, and you could find those short flanking sequences. In human, we're starting to have that kind of power. We now have hundreds of thousands of sequenced human genomes. One problem, though, is that these hundreds of thousands of sequenced human genomes are all siloed, right? So Qatar, the nation of Qatar, is sequencing every Qatari, every ethnic Qatari. 347,000 genomes. The VA in America, the Veterans Administration, is sequencing the genomes so far of 30,000 veterans. They plan to scale to 1 million. Is anyone going to be able to see that data? I don't know. The Qataris can see the Qatari data. Americans see the American data. Chinese are sequencing hundreds of thousands of Chinese genomes, but it's also all siloed. So as of yet, we don't have the resources to detect extremely short coding sequences. We know from brilliant genetic studies, and then lots of follow-up, incredible molecular biology, that there are lethal, if they're lost, protein coding genes as short as 7 amino acids. So a little 7 amino acid polypeptide serves as a neurotransmitter or whatever. There's lots of different types. If they're lost, you die, right? Larval or embryonic lethal. But almost impossible to detect. So unless you get very lucky and get a mutant in that 7 amino acid sequence, you're never going to know it's there. And so now the question is, so we got all these goddamn long non-coding RNAs, how many of them encode polypeptides? Right? How many encode polypeptides instead? So folks have done a lot of deep shotgun mass spectrometry, proteomics. Folks, but there's a serious depth problem there. You get 1 million spectra, you only hit a third of the protein coding genes in the genome. This is too expensive and too slow. A huge breakthrough, which I don't think I have slides on in this slide deck, unfortunately, so I'll just tell you about it. A recent breakthrough was ribosome profiling. Let me make sure I don't. So in ribosome profiling, you try and capture transcripts that are bound by ribosomes, and then you pull those down or otherwise isolate them, and you sequence the fragments of DNA that were presumably in the act of being translated. And that showed, that gave as confusing and as complex a picture of the proteome as RNA-seq had given of the transcriptome. There were ORFs everywhere. Every protein-coding gene had ribosome-bound regions in the 5' UTR, I mean, virtually without exception. Every long non-coding RNA, virtually without exception, had what looked like translated regions. That created a debate that went on for years. The first paper on that, ribosome profiling, was published by Nick Ngoli and Jonathan Weissman in 2011. And they said, okay, everything in the world is translated. Everything's translated. It's all protein. If it's RNA, it's translated. And my group at the exact same time had finished an analysis of shotgun mass spec, and we'd done a lot of statistics, and we said, okay, no, translation's pretty specific. We can't detect any peptides from long non-coding RNAs. And so these papers went out, and it sparked off a debate. Eventually, the initial ribosome profiling result was retracted in Cell. Took 2 years, though, but it was retracted. And they said, okay, no, we analyzed it wrong. If we look at the data slightly differently, we think these other things are ribosome bound. But it's absolutely continued. It's gone back and forth on both sides. And it's still unclear how much of this is translated and how much of this is not. So is it an RNA world with everything transcribed, but maybe only the active transcription, not the transcripts mattering, or is it a short ORF world? And there are thousands and thousands of protein-coding genes that remain completely undetected. There's a study being led by Norbert Perimon right now at Harvard. Norbert has a catalog of 170 short ORFs that were found by really nice computational work that are conserved in every metazoan. Chickens, fish, Drosophila, C. elegans, humans, they're there in everything. None of them have any known function. They have never been hit by any genetic screen in any organism. Right? So what the hell do they do? And now we have CRISPR. Who knows CRISPR? Genome editing tech? Yeah, CRISPR. Now we have CRISPR, and what Norbert is doing is he's systematically CRISPRing every single one of them. And then he's phenotyping not a handful of dimensions, but he's doing many hundreds of dimensions of phenotyping to try and see if there's any change. He's already got a cryptic transcription factor that is only 70 amino acids long, and he's got a new neurotransmitter. Pretty cool. It's pretty cool. So there's a lot of black matter out there that we still don't know what it does. And our classic techniques of, oh, it's lethal, or oh, it's a synthetic lethal. Who knows synthetic lethal? So synthetic lethal is where you're doing a genetic screen. You got a shitload of stuff that's lethal. You know immediately, oh, that's interesting. So set that aside. Now you got a shitload of mutations, vast majority, that have no detectable phenotype. But you've also got a huge catalog of strains of critters what have mutations in. And so you can take them and start crossing them. You can do pairwise combinations so that you start to see every nucleotide together. In yeast, this has been done up to third-order interactions. That's pretty impressive, right? So how many pairwise combinations are there? If you've got 20,000 mutations, what's 20,000 choose 2? Who can do 20,000 choose 2 in their heads? It's a freaking lot, right? It's a freaking lot. It's over 10 million. So, and the third-order interactions are absolutely massive. But these pairwise crosses have been done exhaustively in yeast, extensively in mouse, extensively in Drosophila. I believe now exhaustively in C. elegans by the C. elegans gene knockout consortia. And what you see is a whole bunch of things that had no phenotype when you knocked out one gene, whole bunch, are lethal when combined with one other mutation that also had no phenotype on its own. It's called a synthetic lethal. Synthetic lethal. Who knows the term GWAS? Genome-wide association study? Okay. So when an MD tells you that there's no evidence of pleiotropy in human, well, you know, we haven't done this sort of synthetic lethal analysis in human. You don't get to see every mutation in every genetic background. Who knows pleiotropy? Pleiotropy, well done. You're awesome. Pleiotropy means having different functions in different contexts. So there have now been really, really high-resolution studies of mutation maps. One of the best is by a guy named Dan Jacobson. Dan, by the way, is visiting the University of Birmingham on Wednesday of next week. There'll be an email out. but he's given a seminar at noon in the Center for Computational Biology over in the chemistry building. I highly recommend you attend Dan's seminar at noon. Unbelievable stuff. So he's got mutations in poplar, the tree, that have such high resolution that he's got many mutations in different protein domains of the same gene, in the same gene. So he can ask if effects in, say, a DNA binding domain, if mutations in a DNA binding domain have the same effect as mutations in, say, an RNA binding domain, or so in the same gene. And he's got now libraries of mutants where if the mutation is on this side of the gene, it has a leaf phenotype. If the mutation's on this side of the gene, it has a root phenotype. Right? So it has different functions in different areas of the organism. And that's the problem with multicellular life. So thanks to next generation sequencing, thanks to the ability to see not just one, but thousands of genomes, thanks to the ability to see every transcript in a transcriptome instead of RNAs just one at a time, we're now realizing the enormous complexity of biological systems, which frankly never should have come as a surprise. You have the same genome in the cells that form the cornea of your eye as you have in your nose, as you have in your blood, as you have in your brain. It's all the same genome. It's how it's deployed dynamically across space and time during development and aging that determines function. So should it really surprise us that we have far more genes than we realized when we were looking one cell type at a time, far more proteins and far more functions? It's pretty cool. And that's been the huge boon of next generation sequencing. In fact, now we can start to interrogate the epigenome. Actually, since, when was the first, who knows when the first bisulfite sequencing paper was published? It was quite a while ago. That's 2011, but I think there's one before that. So bisulfite sequencing, so the concept of bisulfite conversion had been around for ages. I mean, at least the 80s, probably the 70s, maybe the 60s, I don't know. But it's a very, very simple chemical reaction, this sulfonation. You take cytosine and you convert it, and the chemical reaction ultimately ends up turning a cytosine to a uracil after it's replicated. But the chemical reaction is blocked by a methyl group, a 5' methyl group on the cytosine. So 5-methyl-C will be unchanged by this, but every single other C in the genome will be changed. At least that's what we tell ourselves. Of course, if there are other modifications to cytosine we don't know about, they may block it as well. But what we tell ourselves is that this changes every unmethylated cytosine into a U. And so now you can do this treatment. You can replicate the genome, in vitro, PCR, and then sequence everything you pull down. And the result of this is that you can get a map. So now you're going to try and map these reads back to the genome. It's a bit tricky, because now you have to treat every single C as either a C or a U when you're doing the algorithm to map it. But you map them back to the genome, they stack up, you see the pattern you use, and everywhere a C wasn't changed in the vast majority of the reads, you say it was methylated. And in fact, this assay is quantitative. So if you're in a complex sample, like say whole animal sequencing, who works on invertebrates here? Invertebrates? Excellent. Which one? Mice. Vertebrate. I think we got to do some more work on your mouse. Yeah. What? That's fabulous. Both are quite awesome. So the thing about invertebrates is when you sequence them, a lot of folks, you take the whole animal, you smush it up. I hope you're not doing this to your mice. You smush it up, and then you just sequence the gamish. And when you have a complex sample like that, you'll get a quantitative methylation profile. If you had exactly one cell type that was diploid, what would you expect? You'd expect every base is either methylated, unmethylated, or hemimethylated, meaning one allele, not the other, right? So it'd be 50% methylated. You'd have half the reads be U, half the reads be C, okay? But that's not the case in a complex sample. So if you're doing mouse brain, for instance, has a very, very complex pattern of methylation, because different neurons have completely different epigenomes. And so this started, this capacity now to see every 5-methyl-C in the genome all at once led us to start to understand the differences between tissues. Because every tissue, during development, ends up with its own unique and beautiful methylation profile. There are many instances of methylation that are the same in every cell in your body. There are some that are in the germline and identical. It's a process called gene conversion, where either the maternal or the paternal allele transmits its methylation status to the other allele. And so you end up with homozygous methylation in both cells. What does methylation do? What's the functional role of methylation in cells? Does what? Stops transcription. Stops transcription. So that's an excellent answer. So when a promoter is methylated, particularly in a vertebrate, it can cause gene silencing. However, when internal exons are highly methylated, it can cause transcriptional upregulation. The most highly expressed genes in most critters, well, we'll do vertebrates and invertebrates separately because they're a little bit different. But most highly expressed genes in vertebrate genomes, and I don't care which vertebrate, I don't care if it's a fish or a mouse or whatever, every single internal exon is going to be heavily methylated. The promoter is going to be demethylated, or would silence transcription, but every single internal exon is going to have virtually every C methylated, the most highly expressed genes. Why is that? And the methylation stops on a freaking dime at the edge of the exon. You can't methylate splice sites for whatever reason. So it's just the exons. It's a mechanistic mystery. The number of mechanistic mysteries in biology right now is exploding exponentially because we can observe all these phenomena, but we haven't yet figured out clever ways of functionally interrogating them. So you guys have incredible opportunities to discover fundamentally new mechanisms in biology because the number of hypotheses now has so far outstripped our capacity to interrogate them that it's just getting embarrassing. And that's a really cool transition facilitated largely by next generation sequencing and omic technologies in science as a whole. And that's the transition away from I have a hypothesis, I'm going to test it, to I'm going to do this assay that's going to let me see the entire functioning of my system, and that's going to build hypotheses, right? It's a fundamentally different paradigm. Instead of hypothesis-based science, it's more like ignorance-based science. I got no fucking idea what's going on. I'm going to see everything and then come up with a hypothesis. So in invertebrates, especially Daphnia, you can have some very beautiful instances of promoter methylation that appear to be highly upregulating. No idea why. But remember that the emergence of CPG islands, who knows CPG island? Very good, very good. Some of you cool kids at the back. The emergence of CPG islands really happened in the vertebrate lineage. And it's when you methylate those CPG islands that you really see silencing. You really see silencing. Whatever the mechanism is that drives transcription out of a CPG island, you methylate that, you shut it down. Is there any reason to believe that there's only one mode of transcriptional initiation? Is the polymerase, the RNA Pol II, you guys seen the crystal structure for RNA Pol II? There's a crystal structure, 1 angstrom in change resolution, huge achievement, absolutely amazing. Got it in several organisms. RNA Pol II has lots of RNA and protein components. Is the structure of that molecule the same in every cell in your body? No, it is not. You have lineage-specific components of some of the most foundational machines, right? So we could have, it's not hard to imagine, that you have different activities, different molecular mechanisms at different elements in the genome. Okay. So I've gone over just a handful of the applications of next-generation sequencing, and I haven't talked about lots of others, and this is a radically incomplete, pointlessly incomplete list. There's a guy at Caltech named Lior Pachter. Who knows Lior Pachter? No worries. Guy at Caltech named Lior Pachter who has a list of what he calls star-seq assays, and I believe that list has now quite a few hundred different assays that are done using next-generation sequencing. It's pretty amazing. All of the sequencing platforms have different advantages, and some are better for some things than others. Illumina, frankly, has outstripped everything now pretty solidly. They can do a human genome for about 800 quid, which is pretty amazing. You guys could think about getting your whole genome sequenced. How many of you do that research thing where they give you like 800 pounds or 1,000 pounds to do research for a semester? Anyone? Is that okay? MRIS program, kids get a little tiny bit of research funding. In any case, you should make your research advisor pay to get your genome sequenced, is my point. It's a good idea. Do it while you're in college. Metagenomics is something I didn't even touch. I've talked as though the world is made of eukaryotes and metazoans. How about prokaryotes? How about bacteria? Who studies bacteria? Several of you, okay. Who studies plants? Okay. All right, so bacteria, sequence bacteria in culture, pretty easy, no problem. Sequence natural populations of bacteria, very challenging. Very, very challenging. What kind of, the bacteria, folks, what do you study? Words. Salmonella. Streptococcus. One critter. One critter. Okay. So you guys don't have to worry about metagenomics. Okay. So if you've got a homogeneous culture, life's pretty nice, not so bad. If you've got something like, say, a metazoan microbiome, say the Drosophila gut microbiome, or the Daphnia gut microbiome, or the bumblebee gut microbiome, how many different species of bacteria do you think you have? You might know for bumblebees. No? Okay. Bumblebees have 4 canonical bacterial species that every bumblebee gut we've ever sequenced anywhere on the planet has in common, and another 4 that are common virtually everywhere. It's work from Nancy Moran, I believe, at Austin, Texas. Incredible work. One of the microbial consortia in the bee gut microbiome forms basically a second epithelial layer. It hugs the tissue, tessellates across it. And if that bacteria is lost, it's lethal to the bees. They absolutely require it. Metazoans have very complex relationships with bacteria. Where did mitochondria come from? Yeah, so lots of folks think that mitochondria are endophytes, right? A bacterial or archaeal organism that had such a close symbiosis with the root of multicellular life that they ended up inside the cell instead of outside the cell. That's why they have their own genome and everything else. In other organisms, we can see endophytes emerging. Who studies aphids? Anyone study aphids in here? A little? Yeah. 8 endophytes and counting, is it now, in aphids? The aphids have a large number of endophytes, of symbiotic bacteria with which they've developed such a close relationship that they now live inside the cells instead of outside. So it's like mitochondria in the making, except radically more recent than evolution. So that's pretty cool. So metagenomic sequencing is where you look at a critter, or say a water sample, or a soil sample, or a piece of plant root, and you sequence the whole gamish, right? With no regard for what's in there. Anyone familiar with tardigrades? Okay, some of you? Yeah. Cute little nematode, also called the water bear. They can go into diapause and suspended animation. They can survive the vacuum of space for a bit. All kinds of cool stuff. Neat critter. When their genome was sequenced, the group that did it reported a remarkable finding that 17% of the genes in tardigrade were bacterial genes. And they thought in the nuclear genome of the tardigrade. That would have been remarkable if it was remotely true. It took less than 72 hours for that to be refuted by Mark, I forget his last name. God damn it, I like that guy. He's at the University of Edinburgh. Okay, a guy named Mark at the University of Edinburgh, very smart guy. Less than 72 hours, he had reassembled the tardigrade genome using more mainstream scripts, actually, than the group that had done it had used. And he showed that it wasn't that they had this huge introgression of bacterial genes in the tardigrade genome. It was that they had failed to clear the gut before they sequenced the fucking thing. So they got the gut microbiome mixed into the Gamisha reads they sequenced, and because their Illumina genome assembler, which is trying to assemble 150 nucleotide reads into contigs, into scaffolds, into genomes, that process had simply not done the best job ever and stuck the tardigrade reads in with the bacterial reads. And they happen to have an especially big gut with especially rich gut microbiome. And so it's just a complete error. Now we know that when you sequence an invertebrate, you get its gut microbiome for free. And folks are very, very careful to handle that well and make sure that you treat the bacterial reads different from the eukaryotic reads. Frankly, isn't so hard because they have completely different patterns of K-mers and things like this. You know what I mean when I mean K-mer? Like 5 nucleotides, 5-mer, 6 nucleotides, 6-mer, etc. Okay, so metagenomic sequencing is pretty cool. Now there's the Human Gut Microbiome Project, right? Sequence everyone's gut microbiome. Humans have extremely complex gut microbiomes, right? Instead of having 8, 10, 20, or 30 species of microbes, we have tens of thousands of species of microbes. Human feces have 10 to the 11th microbes per gram. That's close to a dense packing. It'd be at 10 to the 12th. Absolutely remarkable. Our gut bioreactor we rely on for dozens of services. Vitamin B is nice. Where do you think we get that from? Right? Many species, completely lethal if you lose the gut microbiome. The first one studied really well with termites. Absolutely dependent. Have to have it. Have to have the gut microbiome. Can't make an azenic termite. Some critters have less extreme a relationship. You can make an azenic fruit fly very easily. Can't make an azenic bee. You can make an azenic daphnia. They're sick as hell, very unhappy. They don't breed well, but you can do it. Remarkably, you can make an azenic mouse. You can make an azenic mouse. An azenic mouse is very difficult to breed. There's only a few groups in the world that can do it reliably. But the gut is radically elongated in azenic mice. And presumably that's because they need more space to take up nutrients. So metagenomic sequencing has shown us that gut microbiomes are incredibly complex, very different in every species we look at, very different in different populations of the same species at different places. It's also revealed to us the most complex ecosystem in the world. Does anyone know what is by far the most complex ecosystem in the world? No. Guts are comparatively simple. We farm. We're selective. So when Craig Venter started sailing around and sequencing marine metagenomes, we thought, oh, it's pretty complicated. There's many thousands of things in some of these samples. But then people started sequencing soil, bulk soil. 1 million species per cubic millimeter. 1 million species per cubic millimeter. It is by far the greatest diversity of life ever detected on the planet. And it's not just one soil type, it's most soil types. There are exceptions. There are simple soils. In deserts that have crusts. Have you ever seen a desert that had a cracked surface on the soil crust? Desert crusts are basically biofilms. They're very specific microbial consortia that regulate water consumption in the soil for reasons we don't understand very well yet. They're being studied now by lots of folks. But soil-water interfaces in stream beds have biofilms. Who's ever slipped on a slimy rock while crossing a creek, right? Those biofilms are consortia of eukaryotes, diatoms, and algae, and bacteria. And they're very simple. There's probably a few dozen, probably less, species in there. Not so bad. But when you just walk outside, anywhere, pull up a piece of grass, handful of soil, millions and millions and millions of species. Unfathomable complexity. You have to sequence. Do you realize that if you sequence 1 billion reads, you only get 1,000 reads in each species in bulk soil? Isn't that incredible? So the complexity of that is unfathomable. And metagenomic sequencing has started to reveal it. And that's only the canonical genomes of the critters, right? They all have plasmids, mobile genetic elements, everything else. So metagenomics extremely cool. And plants, by the way, farm and regulate their microbiome just like metazoans do. If you look at bulk soil, right, 1 million microbes per cubic millimeter. And now you start to approach a plant root, the rhizosphere of some plant. I don't know, poplar, Arabidopsis, especially, what do you call them? I don't know, soy. You start to approach the plant root. You go from 1 million microbes to 30,000 for Arabidopsis as you approach the plant root. So the plant is selecting. That's incredible selection, right? It's 10,000-fold selection almost as you approach the plant root. As you go to the surface of the plant root, you can be down to just hundreds of microbes. Plants have very complex communities of endophytes at the root tip, so bacteria that live inside the outermost tissue of the plant root. And then you're at just tens of species for most plants that have been looked at. Right? So that eukaryotes and prokaryotes have very complex symbiotic relationships. These are common to every eukaryote we've ever looked at. And it's pretty cool, and we see it and know it thanks to next generation sequencing. Oh, here's a list of I did have it. This is Lior's old star seq list from a few years back. So there's a shitload of stuff on here. I'll put up the slides you can see. But the proliferation of assays, functional assays that leverage sequencing is enormous. We really have blown way past the day of just sequencing genomes. Right? We can now use next-generation sequencing to interrogate functions like ChIP-seq, like methyl-seq, et cetera. Okay, so that's cool. I'm going to skip some of this. Okay, so let's talk a little bit. We talked a lot about various genomes that have been sequenced and the things we've learned from it. I mentioned in the case of tardigrade that the technology you use to assemble your genome really matters. Let's talk a little bit more about this. So people analogize assembling a genome from short reads to assembling a puzzle. You see these tiny little pieces of sequence, these strings of A, C, Gs, and Ts, and you have to mash them together. So if you've got 250 nucleotide reads and they overlap by 75 nucleotides, you may get a rarely unambiguous matching. But think about the computational challenge of this. If I have 1 billion reads and I want to do all pairwise comparisons of 1 billion reads, that's some serious stuff. That's 10 to the 9th times, it's 10 to the 9th choose 2. Right? That's enormous. It's a quadrillion comparisons. You're talking about serious CPU time now. So the technologies that have enabled shotgun assembly of genomes from short-read, high-throughput sequencing technologies have not been trivial. Getting a nice picture from all the puzzle pieces has not been trivial. Let me see. Is that really all I had on that? Oh, it's annoying. Sorry. Okay, I thought I had something on the actual computational tools. You guys are you a computational crowd? Not a computational crowd. Who codes? Raise your hand if you code as part of your gig. Tiny bit? Little bit? Okay, well, you can talk to me offline. Yeah. So no coders. Okay, so we won't talk about the software. But algorithmically is very complex, and make sure your bioinformatician knows what he's doing. Okay, so Pacific Biosciences. So this was the next huge revelation in sequencing. This company actually started out contemporaneous just about with the rise of Illumina, but it took a lot longer to get the tech working reliably. So they were thinking, you got a polymerase, fluorophores that emit light when they're integrated into the DNA. We have those, had them for a while. Solexa couldn't get this whole sequencing single molecule by termination thing working, but maybe we could if instead of mobilizing the DNA or RNA, we immobilize the polymerase. Who's had a class on quantum mechanics? Okay, you have a physicist, right? So I say zero waveguide, you're right with me. Yeah, zero waveguide. Oh, no worries. Okay. So quantum mechanics, everything's both a particle and a wave. You've got quantum uncertainty. If you immobilize things into a really tight space, the set of energy states that the particle can take on is narrowed, and you can get a very precise signal. So they immobilize the polymerase in a tiny, tiny little space that is not that much bigger than the polymerase itself, and then they just wash a huge gamish of fluorophores for A, C, Gs, and Ts, all 4 bases, so we're not doing 1 base at a time anymore. Just throwing a huge gamish of fluorophores and a huge gamish of whatever the hell it is we want to sequence. And then we just wait for an RNA molecule or DNA molecule or whatever to find its way into one of our zero waveguide wells, get grabbed on by the immobilized polymerase, and then we take a video of the flash. And in this slide, which PacBio showed to venture capitalists to raise $2 billion in less than a year, this slide, you can see this very clean signal rising far above background. Easy to detect. Who thinks the data really looked like that? Never. Okay, so it was about 6 years after this that they could do this reliably. They had reliable sequencing. But it's been really transformative. Anyone have an idea why it might have been so transformative? What's the processivity of DNA polymerase? Who's done PCR? Okay, how long a molecule can you reliably synthesize in a single PCR reaction? Someone, come on, you biochemists? How long? 1,000. 1,000's a good answer. Lots of them have a processivity of about 1,000. Some get up to 8 kb. Some go a little bit longer. So that's why things like if you want to capture an mRNA that's 20 kb, that shit is not trivial. You usually have to break it into pieces, and then you end up ligating them together at the end. It's hard work. The cDNAs are some of the longest genes in the genome. Who knows what the longest genome in a metazoan genome? The longest gene in a metazoan genome. Who knows what it is? Anyone? Neuromuscular junction? Dystrophin. Okay. Very, very, very important gene. Without it, you got terrible disease and death. But dystrophin operates at the neuromuscular junction. In human, it's over 2 megabases. But in just about every metazoan genome, it's the biggest. Getting a cDNA for dystrophin is a total bastard. I think we have one in Drosophila because Sue Selnecker is a relentless human. But I don't think we have one, I don't think we have a full-length one in human. Okay, so this was transformative because although the processivity of, say, Taq polymerase is 1 or a few kb, with these single immobilized molecules in a chemically idealized condition, and remember that this is DNA polymerase, not RNA polymerase, too. So in this idealized condition, you can replicate things that are 100 kb. 100 kb. Pretty amazing. Not always. Usually falls off sooner than that. 10 kb is easy. 20 kb is hard. 40 kb is damn rare, less than 1% of reads. 100 kb is once in a whole run. But it can happen. And so now instead of having 150 nucleotide reads, we're trying to assemble some god-awful puzzle, trying to do massive numbers of comparisons. One run of PacBio, when it first started working really well, you got 22,000 reads, each about 10 kilobases. Well, actually, the first version of technology, 1 kilobase. But the first version that was worth a damn, 10 kilobases. That's amazing. Do you realize how much easier it is to assemble something where you have 10 kb puzzle pieces instead of 150 nucleotides? There's no comparison. Absolutely no comparison. So now you get across an entire bacterial genome with one run at PacBio. You can multiplex 5 or 6 bacteria, get whole genome sequences on a single run at PacBio for about 700 pounds. Amazing. Absolutely stunning. The first metazoan genome that was published using PacBio was Drosophila. And the Berkeley Drosophila Genome Consortia had done 6 releases of the genome. And the 6th and final release before PacBio that came out immediately before the first PacBio genome. And it had pushed the old BAC technology that I showed you at the beginning, the E. coli amplification technology, it had pushed that to the very limit. They had used a brand new optical sequencing technology. Optical sequencing is where you lay DNA molecules out straight on a surface and you're going to film it with a super, super high resolution camera. So you have elongated, straightened out DNA molecules. Figuring out this process was not simple. Like whole chromosomes, okay, laid out. And now, or whole bits of chromosomes. You guys realize how big human chromosomes are, right? You got 3 meters of DNA in every nucleus and every cell in your body. The DNA in your cells is enough to go back and forth to the sun like 7 times, right? It's a massive amount. But they lay out huge chunks of DNA, megabases of DNA. And then they add a restriction enzyme that is highly specific for exactly one sequence. And you put that on, and that is very powerful because now you've got a set of links of restriction fragments. And you can infer where those came from in the genome. And that can allow you to assemble otherwise unassemblable things, like centromeres or telomeres, highly repetitive sequence. They'd done all that. They'd pulled out all the stops. Release 6. Best genome ever, by far, in a metazoan. A megabase of centromeric sequence on either side. Incredible. PacBio, release 7, 2 weeks of computational work, blew it out of the water. Blew it out of the water. No comparison. 200 gaps to something like 10. Incredible. Gorilla has now been done by PacBio. There's a human genome by PacBio. Unbelievable technology. Changed everything yet again. Now we had a view of repetitive genomes. We had no more contigs, no more scaffolds, whole chromosome arms. The first complete centromere has been sequenced now by this technology. Pretty amazing. There's a cute video here, which I'm not going to show you right now, but you can click on it on the slides if you want. That goes through the technology in detail, and I think shows you a video of it happening. It's kind of cool. So here's a read length distribution from the first generation of PacBio that was worth a damn. It now looks better than this. It now looks much better than this. But you see that the mode of reads is still at about a kilobase. But you got a whole shitload, many thousands of reads, longer than 6 kb, some longer than 12 kb. When they did the Drosophila genome, which was 2 generations of chemistry better than this, when they did the Drosophila genome, the longest read they got was 41 kilobases. Absolutely incredible. So the only challenge with this is, maybe I have it on the next slide. Let me see. And there is a big caveat coming. Yeah, okay. The big challenge is the error rate. More than one error at the time the slide was made every 10 nucleotides. That's no longer necessarily true, and that tech, circular sequencing, no one does that anymore. I'll still tell you what it is. So circular sequencing is where instead of sequencing linear fragments of DNA, you circularize them and you put them in, and then the polymerase just goes round and round and round and round. So you see the same fragment many times. And it turns out that if you go around 5 or 6 times, because the PacBio errors are truly random, they are like uniform random. So your chance of making a mistake at any given base is the same as your chance of making a mistake at any other given base. That's pretty cool. So if you go around 5 or so times, you're going to have perfect 99.9% error-free sequence. But no one does that anymore because the long reads are more valuable. No one gives a shit about the error rate because computers are faster. But so the error rate now is about 1 base in 15, 20, something like that. The downside is, with this technology, do you think that the errors are insertions and deletions or base substitution? Just a guess. What's your intuition tell you? Anyone? So, little column A, little column B. But deletions are the real bastard because you just miss one of these flashes. It doesn't go quite bright enough. Your camera doesn't quite detect it. As a result of that, they turn up the yield on the cameras so high that actually one of the main types of errors they make are insertions, calling a base that wasn't there, thinking they saw a flash when they didn't. And that's tough because the search space for insertions and deletions is exponential, right? Not linear. So that's tough. So mapping these reads is expensive, but there are now some great computational tools out there. So it's pretty stunning. It's pretty amazing. Virtually no one, well, no one with sense, sequences a bacterial genome with anything other than one of these 3rd generation technologies. You don't do it with Illumina anymore. There's no point. Why go through the heartache of trying to assemble the reads? You just do one run on this. You have a perfect, error-free, single contig circular genome. Okay, so PacBio is awesome. And it also, there's a really neat thing I'll talk about in a minute with DNA methylation. But so PacBio has now blown everything else out of the water for resequencing whole genomes. Like Ion Torrent has, plus forget that, it's an old slide. No, PacBio is what you do now to sequence whole genomes for a whole genome you've never seen before. So, Daphnia may cost you 30 grand because it's a 200 megabase genome, but a 5 megabase bacteria is going to cost you virtually nothing. Right? It's going to cost you less than 1,000 pounds. Okay, so the other thing is DNA methylation. Because this, God damn it, because I'll just tell you about it. Because this technology is sequencing by replication in real time, do you think it takes the same amount of time for DNA polymerase to attach a base and have base pairing with a methylated C versus an unmethylated C? Think it takes exactly the same time? You think quantum mechanics exactly the same? Shit, no. If there's a modification on the native strand and you're amplifying it and sticking in a new base, if the native strand has any covalent modification on it whatsoever, it will cause lag time in the replication. Lag time. 6-methyl A and 5-methyl C have different lag times. Pretty nice. So if you know exactly the signature of the lag time created by a covalent modification to native DNA, you can call not just a 4-letter alphabet, A, C, Gs, and Ts, you can call a 5 or 6-letter alphabet with the covalently modified nucleotides. And that's been really transformative too, because now you don't even have to do bisulfite sequencing, right? You don't even have to do methyl-seq. You just sequence it on a PacBio, and you read off the native modification platform. But there are challenges with that because you have to learn what the lag is, right? You have to know, okay, the lag time was this long, not that long, therefore it's this, not that, right? And so it's a little bit challenging, but like I said, people have got it up to at least a couple of modifications. So that has helped plummet the cost of sequencing even further. so PacBio genomes, yeah, absolutely stunning. Absolutely stunning how much you can do. Bacterial genomes per dollar. What are weird units? This is a shit slide. I hate this slide. Okay. It's bloody cheap. So you multiplex, say, 6 bacteria onto one PacBio run, you're probably going to have single contig genomes for every single one of them. So that's about 120 pounds a genome. Pretty cool. Okay, that's let people do some really cool stuff. Nick Lohman here has used some of these technologies to do epidemiology. So now you can do epidemiology in bacteria. You can do things like track the spread of an epidemic. So you can have not just one bacterial genome, but say one bacterial genome from every single patient with a disease. And you can form the complete epidemiology of the transmission of the disease back to patient 0 based on SNPs in the bacterial genome. Pretty amazing. They did that for Ebola. They did that for Zika. They did that for Lassa. They've done it for food poisoning in Seattle. It's been pretty cool stuff. Okay, and let me see. But, screw that. How much time do we have? Oh, this was cool. This was Nick's first big thing in 2008, an outbreak. Do you guys know Nick Lohman? He's here. Go bother him. Find him in the hallway. He's awesome. He's a rock star. Talk to him. He's done some really cool stuff. So he managed to track an outbreak by SNPs. And let me see if he's... Oh, this is so cool. And so he traced it. He traced the outbreak to a specific unit in the hospital and then a specific bed as patient 0. It's pretty neat. And so he got the whole thing by doing bacterial epidemiology. And since then, he's done this at radically higher scale. I guess I don't have those slides. He's done this at radically higher scale with an Ebola outbreak now, where you get every patient, across many countries, and, it got to the Gold Coast here. Incredible, incredible stuff. It's, is it useful? I don't know. Are there applications? But lots of Centers for Disease Control are funding this stuff because probably the applications are going to emerge. People are using it in cancer biology to understand where the origins of cancer resistance. You can sequence a shitload of cells from one or many tumors and understand essentially the phylogeny, if you will, of the cancer cells by their patterns of mutation. Old mutations versus new mutations. Pretty cool. And that is, for the first time, by the way, that has really, really started to be transformative for cancer biology. The first sort of molecularly informed drug actually was just approved. So lots of cool stuff. I'm going to fly by that, though. Yeah, and of course, most chemotherapies are DNA damaging. So you give someone a DNA damaging agent, now you sequence and everything's shredded, but you can still track the evolution. And there's been tons of just incredible stuff on this. So that pushed by really two fields. One is cancer biology, and the other is bacteriology, microbiology. Saw the rise of single-cell sequencing. So now for the first time, advances in microfluidics and then nanofluidics gave rise to the capacity to capture a single cell and make a DNA or RNA library from a single cell. So now you can take a bacterial colony and ask questions like, is there transcriptional or genetic diversity in my bacterial colony? Absolutely stunning stuff. And so people learned about how transcriptional initiation is a stochastic process. They were able to fit different types of models to it, got some nifty mechanistic insights, particularly from Adam Arkin's group, particularly in microbes, and some pretty incredible stuff. And now Howard Chang at Stanford, lots of other folks have done amazing applications of single-cell sequencing in many, many systems. And there's some very tricky biochemistry, right? Because if you got a single cell and it's haploid, you have exactly one copy of the genome. And that's tricky even for PacBio. So people invented these branching amplification processes called whole genome branch chain amplification. And that gives you, from one copy, many copies of the genome. But of course, it's very spiky. You have exponentially more coverage in some areas are deserts. It's tough. But you could do it. So you can get a view of what individual cells are doing and what their genomes look like. And so for cancer cells, now you really could trace every cell and know where it came from, where's its lineage, where's it's from. Okay, so yeah, PacBio. Oh, there's the zero waveguide slide. That's pretty. That is what the wells actually look like, that cartoon of the wells. Okay. Now, PacBio was doing great, thinking, holy shit, we're the only guys doing single molecule. We've got reads of unprecedented length. We're pushing our prices down with advances in chemistry. We are going to rule the world. Enter nanopore sequencing. So nanopore sequencing is called Oxford Nanopore is the leader, but the key technology was invented at UC Santa Cruz in California. And there are two key advances. One was an advance in essentially synthetic biology / material science, that you could embed a protein pore in a semiconducting chip. And that was incredible. Now, once you've done that, you could imagine passing a biopolymer through that pore and reading it off by passing electricity across the semiconductor and looking for changes due to what's going through the pore. And so you get a femtoamp signal as the electricity passes through. Very, very low level, but very, very detectable and very, very measurable, just like the ion torrent technology. Nano, pico, femto. Tiny. Okay, so this thing's going through, you're reading off a change in electrical current, so it turns out that A, C, G, T, what the hell else ever, will create a distinct electrical signature. And I'll show you some real examples of that in a minute. That's pretty amazing. So now the next major technical challenge was, okay, how do we get it to flow through? Well, of course, you could have a protein itself that has ratcheting action, like someone name a protein that has ratcheting action. I'll be very impressed. Several excellent right answers. Yeah, absolutely, locomotor proteins. And how about polymerases? Right? Polymerases chug along. How about exonucleases? Exonucleases could be used to create a very precise signal because they're going to chop off one nucleotide at a time into the pore. And so all of these technologies were explored. But the key thing that they really needed to make this rock and roll was they had to have a nice gradient so that they could basically do gel electrophoresis from one side of the pore to the other to help everything keep getting sucked into these pores and pass through. Now, to do that, they needed, and to have all these biomolecules be nice and stable and readable and, in a nice fluid mode, not a gel, they needed really high saline, really high saline. Vast majority of name your favorite biomolecule do not work in incredibly saline environments. So they went to the salt pits in the San Francisco Bay, where they actually just dry out seawater to make sea salt. There's stuff living in there. There's all kinds of bacteria, all kinds of life, extremophiles. And in that gamish of extremophiles, they found various types of pores with various locomotor proteins that could tolerate the levels of salinity they needed and beyond. So they founded a company, Oxford Nanopore, for various reasons, and this thing was off and running. This is a mini ion sequencer. When Nick gives this lecture, he brings one. I always forget. But it does. It fits in the palm of your hand. It's a USB drive. Until you have pipetted into your laptop, you have not lived. So it sticks into your laptop. You open the thing. You squirt in the library. Library preparation now takes 15 minutes. There's 2 steps. A monkey can do it. I can do it. I'm a computational guy. I can't do anything. But absolutely stunning. So the reads get sucked through. At the time this slide was made 2 years ago, 100 KB reads. Now, megabase reads, megabase. The throughput otherwise is about the same as PacBio, but the error rate is still higher. And there are still challenges. This is not perfect yet, and it has not killed PacBio. PacBio has their error rate down to about 2% now in the brand new machine. Does anyone know, out of curiosity, what a PacBio machine weighs? First generation weighed 2 tons. Current version weighs a nice light spry 1 ton. So in most labs, you have to have the goddamn floor reinforced to bring one in. This is a thumb drive. So when Nick does bacterial epidemiology in Brazil or Africa or where the hell ever, this is what he brings. On his Ebola expedition, he had competition from another university, and they had brought an Illumina sequencer, which is, yay big, several hundred pounds, right, and trucked the thing into the heart of Africa. And of course, Nick's sassy students came with their, little lunch bag of sequencers. Okay, so they have a big boy version of this too. They have several big boy versions of this, but the big versions are nothing but aligned stacks of those little flow cells. There's a video link you can find in the slides. You can go look at this technology happening in real time. It's pretty cool. Here's a schematic. This is the slide that Oxford Nanopore showed to investors when they were raising their billions of dollars in initial capital. Who thinks it looked like that? No, my God, no. So it's actually, this is sort of how it looks in character, but not in reality. When it's sucking through the pore, it ain't one nucleotide in the pore. Nucleotides are really tiny. It's about 5 nucleotides in the pore. The electrical sequence that you're reading is not caused by an A, a C, a G, or a T. It's caused by AATAA, or whatever, a sequence of 5 nucleotides. So the alphabet is not 4 things, it's 4 to the 5th. It's a big alphabet. That's a big multi-class classification problem. So base calling was not easy. Many generations of this technology, you could see signal shifts, and the signal shifts really were big. That's not a lie. They were big, but they sure as hell didn't look like that, and you sure as hell couldn't figure out what the sequence was from them. So it was a pipe dream for ages. They announced that they had solved sequencing before they could call a single base. They were arrogant, crazy assholes. And there was an 18-month period where everyone thought they were freaking liars. Literally, 18 months. And then they said, sequence! We weren't lying. Right, 18 months later, after announcement. But, and Oxford Nanopore has made a history of that. They've made huge announcements, at least a year in advance of actually being able to deliver what they could do. And that's created a fascinating public perception. But now, for the most part, all is forgiven because the goddamn thing works. This is real data. This is showing several replicates of the same sequence. That is actually what it looks like. However, keep in mind it has been scaled and squished. In reality, every single one of those peaks will have a random length. So you have random transit times through the pore that we don't yet understand. Probably inhomogeneities in the gel or the electrophoresis or who this shit knows. But the transit times are random. So that signal looks much, much messier. And you can see that even with all the effort we've done to normalize it and squish it so that every event has the same length, there's enormous variability in what the signal actually is. But at the same time, you see that nice tight black line on average. It's pretty good. You don't have too much of this nonsense. It's pretty good. And that was over a year ago. In reality, there's quite a lot of variability, but with clever normalization methods, you can tighten it up pretty good. And now we've done things with it like sequence the Drosophila gut microbiome. This is from my group in collaboration with Susan Selnecker. We've got a great algorithm that we built in collaboration with Nick Lohman and some other folks here at the University of Birmingham. We used it to develop an assay to detect, we call ModSeq, to detect not just a single class of covalent modification, but absolutely any covalent modification to DNA. So now and the wonderful thing about Nanopore, with PacBio, you gotta, look at a quantitative lag that has its own mean and standard deviation. With Nanopore, every covalent modification has a pretty goddamn distinct transition in the electrical signal. So you just have a different step function. And so we can read off the covalent modifications, even if we have no idea what they are. We can say, okay, this base was modified, this base was modified, this base was modified. If the only modification in your genome is 5-methyl-C, it means you've got a complete methylation map. If you have more complex landscape of modification, you may not necessarily know. But we've done some cool shit with it. It's pretty amazing. We can really reliably detect some types of modification, not so much others. They're now developing methods for doing protein and RNA sequencing. And here are some more stuffs for you. That's it. So I'm going to end here, take a few questions, and then I got to run because I have go give an exam to a bunch of poor bastards. But that's a rundown. That's where we are now. PacBio, Oxford Nanopore, now dominating genome resequencing. Oxford Nanopore on the verge of dominating epigenetics. Absolutely stunning technologies. The prices is dropping. The prices of both of them are dropping continuously, and they're finally starting to force Illumina down. And it has created a landscape for incredible scientific achievement. So if you've not already embraced next generation sequencing in your education or research, I highly recommend that you do. It's incredible. Questions? Yeah.
If you're looking at a specific set of genes, a very small set, like 20, and you want to do methylation checking, is bio-sulfite sequencing still the way to do it?
What kind of genome are you in?
So it's just for our project, it's for mice.
I see.
So we do mouse genomes, so they're pretty well identified already.
I see. The mouse can be, yeah, that can be a bit tricky because you got 20 genes in a massive genome, so you probably don't want to do the entire genome, right? And so I would recommend probably something, capture-seq. How are you doing it now?
So it's not, it's a hypothetical thing, but we've got a week more than these guys.
So I think you probably want to design probes to your 20 genes. Tile them with bioattenuated probes. You can buy those, but they take a couple weeks to arrive, so you probably want to order it. And you can just do a streptovat and pull down on those, and then do bisulfite sequencing on the resulting radically reduced library. Right? That way, with one lane of HiSeq or one MySeq run, you'll have pretty rich characterization. Remember that you have to aliquot your sample because part you do bisulfite, you need the control.
Would I be able to email you it for it later, for like a thing that I can write down?
What's that?
Would I be able to email you it for it later?
Oh, yeah, of course. Yeah. Other questions? Okay. In that case, I am going to wrap it up.